Morning. Thank you to the organizers, especially to Elizabeth Polliser for organizing this event in such troubled times. Our section at the German Environment Agency is focusing on toxicology and health-related environmental monitoring. Two main German human biomonitoring studies are located at our section, and we are as well a coordinator of the HBM4EU program with the goal to coordinate and advance human biomonitoring in Europe. In other words, we are experts in human biomonitoring. But what about gender? The development of the gender mainstreaming in our house brought us to the awareness that we need to take it into our consideration. We were also willing to do so, but our problem was the absence of handouts, ready-to-use tools and approaches. In this context today, I will present to you our contribution to a research project, that is, integrating sex-gender methods into human biomonitoring studies. That should find a remedy for our problems. I will turn off the camera for the rest of the presentation to give the opportunity to concentrate better on the content. I will give you an overview of the joint research project INGA. Then I will provide a short definition on human biomonitoring and evaluate the state of the art of sex and gender incorporation in the HBM surveys. Afterwards, I present the INGA proposal for a sex-gender-oriented questionnaire for environmental health studies and our first results of its application. Last but not least, conclusion and the outlook. The project INGA stands for Integrating Gender into Environmental Health Research. It is a third-party funded project supported by the German Ministry of Education and Research. All partners are engaging the sex-gender approach in different fields of environment and health research, such as, besides the human biomonitoring, epidemiology and public health, and are supported by the gender studies. In order to give recommendation to the public, the politics or in science for implementing sex-gender methods, we have analyzed the state of the art, built a scientific basis, and evaluated and tested methodology that could be recommended for sex-gender assessment in particular disciplines. In human biomonitoring, we are determining the concentration of chemicals and their metabolites in tissues like blood and urine, and on this basis, we are making conclusion on human exposure. Biomonitoring data directly reflect the total chemical body burn resulting from all routes of exposure and inter-individual variability in exposure levels, metabolism, and excretion rates. The sex-gender perspective, on the one hand, stands here for the biological factors that can determine the intake and metabolisms of the chemicals. And on the other hand, the behavioral and lifestyle factors, for example, sports, food consumption, or living environment, that primarily would influence chemical exposure, but might also act as modulators for the biological factors fitting well the embodiment approach. To identify the potential for sex-gender analysis within the human biomonitoring, we performed a systematic review of the questionnaires used in the HBM studies so far. In the first screening approach, 34 human biomonitoring studies all over the world were selected. Since we wanted to reflect the questionnaires, the first selection criteria was availability of the questionnaire in German or English. To pick up the actual status, the survey year after 2010 was included. As additional criterion, the characteristics of the study population was applied. For example, mere mother-child pair studies were excluded since half of the, for us, interesting population is missing, and studies with small numbers of participants were taken out. So 5 international relevant HBM studies and their questionnaires have been identified and analyzed in details. These are the 5 detected relevant HBM studies from Canada, Germany, the United States, France, and the UK. The first 3 studies are representative cross-sectional studies, and the current available survey cycle has been used for the review. The longitudinal cohort study of children ELFI from France has been included since they have interviews for the mothers and fathers. so the family as a system is captured. Other cohort studies of children rarely or didn't include the fathers at all. And at last there's the UK Biobank, a population-based cohort study that collects urine, blood and saliva to archive for future analysis. For this comparison, all the differences in the study design and the target groups, if children or adults, have to be considered. The systematic reflection of the remaining questionnaires follows the sex and gender-based classification of Nancy Krieger and the multidimensional sex-gender concept developed by the INGA project, with the gender dimensions, sex assigned at birth, current sex-gender identity, internalized sex-gender roles, and externalized sex-gender expressions. So first we look at the sex-based variables like height, weight, hip circumference. But since HBM studies have a medical examination, these basic variables are, as expected, included in the surveys. More complicated is the answer for the sex variable. Even the simple, just binary categorization men-women is collected differently. In the reviewed studies, the information is taken from public authorities or from the interview with the participants. But there are also indications that in some cases the interviewer is taking this classification based on their own impression. And there was one study that also had the sex by chromosome determination. All these informations are used for this one variable men-women, even though the underlying meaning can be totally different. This shows how important clear definitions and standards are, even for just the comparability of the data. However, the socialized sex-gender or gender-based variables were hardly found in the questionnaires. So the questions to how do I see myself or what role models are anchored in me and my environment were absent. With the exception for the French study Elfie, where the dressing preferences for the babies was included. To sum up this part, a lot of variables are routinely collected in human biomonitoring that can be used for the sex-gender analysis, but they lack of standardization. A new questionnaire for environmental health studies that is currently under development by INGA anticipates to address these ambiguities and gaps. We are fortunate to have the German Environmental Specimen Bank ESB settled at our department, so we were able to test this questionnaire in 2019. The ESB analyzes the exposure of humans and the environment to chemicals in different ecosystems. Since 1985, they have been collecting thousands of samples every year to store them permanently. In the human part, regular standardized sampling of about 600 students at 4 locations in Germany is carried out yearly. The test subjects receive self-administered questionnaires on behavior and living conditions, the 2 status is recorded, and urine and blood samples are collected. The additional questionnaire based on our INGA concept included a total of 33 questions concerning sex-gender instruments. And our goals were, on the one hand, to test how the questions are accepted and understood, and on the other hand, how to create possible alternatives to the binary sex-gender consideration. 513 students took part in the survey. In the relation to the gross sample, we were able to realize a subsample of 505 students that handed in our questionnaire on top. That is an excellent response rate of 98.4%. This shows on a first step that there is a high acceptance and low inhibition threshold towards those kind of questions in this population group, and that the €5 incentive was doing its job. So we were left with 502 usable cases in the net sample, since 3 students filled out all the questions except the last ones concerning their gender, and therefore, based on the case definition, we excluded them. We decided to use the 2-step question method for assessing gender categories developed and evaluated by Charlotte Tate and colleagues, and asked the students first to identify their current sex-gender identity and then the sex they were assigned at birth. The two-step gender question will help the precision of measurement crucial to answering the fundamental questions posed within each domain. The answers of the current sex-gender identity show even in this small sample a variation. This means it can differ from the sex assigned at birth. Two persons answered different identity not mentioned here, and one did not want to be categorized at all. The second question, what sex were you assigned at birth, illustrates the attribution of others, in this case the medical staff at the hospital, and the binary answer possibilities was modified based on the jurisdiction with the third category intersex. But since the students are in the age range of 20 to 29, the third category was still missing when they were born, so it makes sense that no one chose this possible answer intersex. Now I will show you results for tested instruments to investigate the missing dimensions of internalized sex-gender roles and externalized sex-gender expressions. We have designed several questions regarding internalized sex-gender roles, whereby in this context I will only deal with the revised BAM-sex role inventory. The original BAM-sex role inventory was developed in 1974 by the psychologist Sandra L. Bam. The test subjects should use a list of previously determined socially desirable gender-typical characteristics, such as warm, sensitive, or dominant, authoritative, to indicate how much each characteristic applies to them or not. As part of the German Environment Specimen Bank, we used the revised German version by Troche and Ramseyer from 2010. They tested the instrument on students in Germany, and based on the collective, they updated the queried personal traits. For visualization, we ordered the masculinity traits on the left side of this network diagram and the femininity traits on the right side. The red and blue circles are the connected mean values of the personality traits separated by the biological sex. It shows graphically that in average, the female students rate themselves higher at the femininity traits. On the right side is the blue line above the red line, and the male students vice versa. On the left side is the red line higher than the blue line. This direct visual comparison shows that women and men tend to claim the personality traits overall similarly, but in different intensity. We need to split the sample to generate classifying scores that can be used for the gender roles in the next step. For the dimension externalized sex-gender expression, I show you the results of the SAGE scale on our sample, because this question battery measures external perception, or rather the conformity of the gender expression expected from the outside. Students should classify themselves on a scale what they think others would say concerning their looks and behavior, from 1 very feminine to 7 very masculine. Depending on the sex assigned at birth, a strong conformity group can be generated. Means if female students, for example, see herself as very feminine in appearance and behavior. Moderate conformity are the students that tend to place themselves in the middle of the scale. And then there's a group of non-conformity. Those students did not comply with the externally expected gender roles. What do the results show us and how can we use them? First of all, there's a change over time in the classification of the current sex-gender identity compared to the sex assigned at birth. And therefore, we think it is important to integrate the two-step question as a standard. Even though it is not possible to generate a third group at the Environmental Specimen Bank due to the small sample size, it helps to precise the measurement. In our opinion, the revised BAM-sex role inventory and the SAGE scale are promising tools for the HBM. They might capture the missing dimensions, and by going beyond the binary of the biological sex, they can help to explain other underlying mechanisms. In the next step, we want to apply those instruments on the chemical exposure assessment in human bodies. But since the clinical parameters have to be analyzed, there is at least a year between the sampling and the available dataset. And so we will hopefully have first results ready concerning the chemical exposure for the next Gender Summit. Thank you for your attention.